classroom here. I made a post yesterday on our classroom about a tutoring opportunity that is going on tonight for anybody that is interested. So right after this class is over at 6:30, I host an advisory class that we do different stuff different weeks, but this week on Thursday today, the other teacher, Miss Mota, and I am going to be offering math and science tutoring for anybody that is interested. So if you have any questions about any of your math class or any of your science class, biology questions, or if you're taking the Algebra 1 star or the Biology star, then you're more than welcome to stop by and get some help, get some tutoring, whatever you need. It's from 6:35 to 8 p.m. So anytime within there, feel free to come by and jump in with us, ask questions that you might have about this class, about other science classes or other math classes that you are a part of. So that's going on tonight after we're done with this class from 6:35 to 8 p.m. The link is posted right here on our Biology B home screen. If you scroll down a couple posts, you'll see my Science Tutoring Thursday link. So feel free to jump on there between 6:35 and 8 o'clock tonight if you want some extra math or science tutoring help. All right. So getting into our notes for tonight, we're basically going to be continuing on with the notes that we were talking about yesterday. So I'll do kind of a brief review of all the stuff that we were going over on Tuesday, and then go through some of the new stuff that we're going to be talking about on today, Thursday. So we started off last week talking about chromosomes, these tightly bundled and packaged segments of DNA. In humans and a lot of other animals, we have what are called homologous chromosomes, which just means that we have 2 copies of each type of chromosome. So humans have 23 different chromosomes. So we have 23 pairs of chromosomes. One that we get from our mom and one that we get from our dad. So the chromosomes have the same genes on them. You inherit the same genes on the same chromosome. And whenever we go to copy our DNA and divide, we make a copy of that chromosome, and that's called a chromatid. So you have the one chromosome from your mom and dad, and then right before the cell divides, it makes a copy of them. So now you have 2 copies of each one of your chromosomes. And we call those copies that we made chromatids. There are 2 different kind of sets of cells that we have in our body: somatic cells and gametes. Somatic cells are like your normal body cells, like your skin, muscle, brain cells. And these guys are diploid, meaning that they have that normal set of 2 copies of every single chromosome. The other, like 1% of cells in your body are your gametes, your sperm cells or egg cells. And these guys are a little bit different. These guys are not diploid, they are haploid, meaning that they only have 23 chromosomes inside them. They only have 1 set of chromosomes. So again, diploid, double set of chromosomes, which is the case for like 99.9% of the cells in your body are diploid. But that last little bit, those gametes that we have, those are haploid cells. So they only have 1 set of chromosomes. We're going to be talking a whole lot about these guys today. So get ready to hear a lot about gametes and a lot about haploid cells. Hey, Samyan. Good to see you. Welcome.
Hi, how are you?
Hello, hello. Doing good. So in humans, we have 23 different chromosomes. And so most of our cells that are diploid have a total of 46 chromosomes. So 23 pairs of chromosomes gives us 46 total. But in those haploid cells, in those cells with only the one set, there's only 23 total chromosomes. So most normal cells have 23 pairs, giving us 46 total. But our haploid cells are those gametes, they only have 23 chromosomes. They only have one full set of chromosomes, whereas most cells have two full sets of chromosomes. Talked about some issues that can happen if the chromosomes don't line up correctly. We then talked about cell division, and in most cases, it's a way to repair or replace cells. So if we need to make new cells, or if cells got damaged and we need to make new copies, that's where cell division comes in most cases. We went over the cell cycle, where we had that G1 phase where the cells are growing like normal, everyday type stuff. And then if they need to divide, they'll first go through S phase, where they copy their DNA. Then they'll go through G2 phase, where they copy everything else in the cell. And then they go through that M and C phase, the mitosis and cytokinesis phase. And this is where the cell actually splits apart into two pieces. So the main type of cell division that we focused on yesterday was called mitosis. Mitosis produces two identical diploid cells. So both of these cells have two full sets of chromosomes, and they are both identical to our original cells. So this type of division is what happens in your normal body cells, those somatic cells that we were talking about. So you start off with one, and then you may basically make an exact carbon copy, and you end up with two new cells. So we start off with prophase when we're getting ready to actually go through mitosis. Prophase is our preparation phase, where the DNA bunches up really tight and you can really see the chromosomes, and the nucleus starts to disappear. And those asters and spindles, those things that are actually going to pull the chromosomes apart, those start to form and start to move into position. So again, prophase is the prep phase. Everybody's getting ready. We then go through metaphase, or the middle phase, as I like to call it, where all of our chromosomes are lining up right across the middle of the cell, sometimes called the equator of the cell. And the spindle fibers are attaching to our chromosomes. So we're getting ready to get pulled apart. So we're lining everything up just right, because up next we have our anaphase, our action phase, where the chromosomes get separated. They get literally pulled to either side of the cell by those spindle fibers. So we have those two copies of each different chromosome, and then those different chromatids, they're then pulled apart by the spindle fibers, and one full set of 46 chromosomes goes to one side, one full set of 46 chromosomes goes to the other side. So we're separating them apart from each other, and we're pulling them to opposite sides of the cell. That's an anaphase, the action phase. We then have telophase, where everything starts to kind of come back together. So the DNA becomes less tightly scrunched up, and the nucleus starts to reform around our chromosomes. And then finally, at the very, very end of everything, we have that cytokinesis, where we do that final little pinch apart and separate the cells so that they become two distinct new cells. So we, before this, had one cell that was one big cell with two sets of chromosomes on either side that was still attached by like a little piece in the middle. Cytokinesis is where we snip that piece and the two cells become individual, brand new, identical diploid cells. So we start off with what's called the parent cell, the original, the OG cell. We go through prophase, the nucleus starts to disappear, the spindle fibers start to form. Metaphase, everybody lines up right across the middle, attaches to those fibers. Anaphase, everybody gets pulled apart to opposite sides of the cell. And then telophase, the cells become separated almost, and that nucleus starts to reform. And again, at the end of mitosis, we have two cells. So we start off with one cell. We end up with two cells. They are diploid. They both have that full set of double chromosomes, one from the mom, one from the dad. And they are identical. So both of these cells are identical in form and function to each other and to that original cell up here. So in cell division, we call it parent cells and daughter cells. And so we can say that mitosis produces two daughter cells that are identical and are both diploid. So whatever we started off with up here, we essentially now have two exact copies of that cell down here. So that is mitosis. And remember, the way I always remember mitosis is because mitosis has this TO, like 2 in the middle. Mitosis makes two cells. All right. So that's great and all. It's awesome. This is super helpful for making new cells, repairing and replacing cells. But what about if we don't want to make identical cells? What about if we want to make something that is a little bit different, that we need for something else besides just repairing and replacing? In that case, we have to go through not mitosis, but meiosis. So meiosis is a very specialized version of cell division, because meiosis is what produces our gametes. So in humans, we have sex cells. So if you're a male, you produce sperm cells. And if you're a female, you produce egg cells. And so meiosis is what produces those cells. Because remember, these gametes are not like other cells in our body. These gametes are haploid. They only have one set of 23 chromosomes, not two sets to make 46 like all the other cells. So meiosis is also different because we don't make two cells, we make four. So we start off with an original parent diploid cell, similar to mitosis. But at the end of everything, we have four haploid cells. So that is the 2N. You'll see the N's used a lot. 2N just refers to being diploid, having two sets of every chromosome. Single N refers to being haploid, having just the one set of every chromosome. So this is different. We're not just copying and pasting the cells. We're making different types of cells. We're ending up with these haploid cells. So we're cutting the chromosome number in half for each one of our new cells. So the reason why we need meiosis, you may be wondering, well, why do these gametes have to be haploid? Why can't they just be diploid and these go through mitosis just like everything else in the body? Well, there's a very good reason for that. And that is because these gametes are what are going to be used to produce a new baby, right? In order for a new baby to come along, you have to have a sperm cell and an egg cell meet up and fertilize each other. So we know that at the end of the day, we have to have a new cell that has 46 chromosomes in it, right? Eventually, that's what we need to produce all the other cells in our body. We're supposed to have 46 chromosomes in most of our cells. But if our sperm cells and our egg cells both were diploid, both had 46 chromosomes, when these guys come together, they combine their chromosomes. This is why you get half from your mom and half from your dad. So if this guy was 46 and this guy was 46, we'd end up with a new cell that had a combined 92 chromosomes. And obviously in human cells, that does not work. We talked on Tuesday about having even just one too many chromosomes is a really bad thing. So we need to make sure that when this sperm cell and when this egg cell meet up with each other, they'll join 23 chromosomes from the dad, 23 chromosomes from the mom. They'll fertilize, and that's what will give you your zygote, which is your special fertilized cell, that now has a total of 46 chromosomes. So we have to make sure that we have two haploid cells come together so that you can have your dad's set of 23 and your mom's set of 23 join up and produce a new set of 46. So then when the son or daughter grows up and is getting ready to have kids on their own, you start off with a diploid cell in either the testes or the ovary where these are made. But then that has to produce haploid cells so that they can meet up to produce a new diploid cell. And the way that we make new haploid cells in our body, that's right, it's meiosis. So these guys start as haploid, but eventually will come together, fertilize, and that will create a new diploid, a new cell that can then go on to produce all the other cells that our body needs. So if you have an egg that has a single set of chromosomes and a sperm that has a single set of chromosomes, you'll end up with the zygote that now has a double set of chromosomes, just like it's supposed to. So we need meiosis to cut down on that chromosome number and make sure that our sperm and egg cells are all haploid, all only have one set of chromosomes. Does anybody have any questions so far? Anything about all this like haploid, diploid stuff that you're still a little bit confused about?
No.
All right, awesome. Grab some more water. All right. So we need to make these haploid cells. And I mentioned earlier, we have 4 cells that we end up with. So instead of just dividing our cell once, like we do in mitosis, in meiosis, we actually go through 2 full cell divisions. So I basically copy the DNA, split it up once, and then split it up again. And that second split is where we end up with that 4 haploid cells. So the first part of meiosis looks a lot like mitosis. The DNA gets copied, the cell splits apart, and then we end up with two diploid cells. So after meiosis I, we still have two cells, and they're still diploid. They still have the same amount of DNA as the original parent cell. There's also something, so that part is very similar to mitosis, but there's also something in meiosis that happens that's a little bit different from mitosis, and it's called crossing over. So during that first part of meiosis, those homologous pairs, those two original chromosomes from your mom and dad, make their chromatids, so they double up with each other. They then line up in a certain way. They all kind of line up right next to each other, forming what's called a tetrad, because tetra is kind of like another version of saying like four. So a tetrad, because now you have four chromatids that are all lined up real close to each other. And a funny thing happens in meiosis when this lining up happens and they get close. Sometimes part of one piece of DNA will accidentally get crossed over with another piece of DNA. Remember, these are they're still all the same chromosome, so they have all the same set of DNA on them. So essentially what happens is that the strands accidentally line up with the chromosome next to them, not with each other. And we call that crossing over. So sometimes when that happens, you can end up swapping genes from one side to the other. So it's all the same genes, but your mom's genes and your dad's genes might have like a little bit different versions. Like maybe your mom's gene has the gene for blue eyes and your dad has the gene for brown eyes. When that crossing over happens, they can actually swap around some of their genes. So it looks kind of like this, where you have your mom's chromosome and your dad's chromosome, right? So here's our diploid cell, two copies of the same chromosome. And you'll notice that they both have ABC genes, but the mom has the little version of ABC and the dad has the big version of ABC. When we get to talking about genetics and how genes work, we'll talk about why there's big and little ones. But for now, you just have to know that they're the same gene, but different versions, essentially. So we have that diploid cell. We then go through that DNA synthesis, and we now have copied our DNA. We have 4 copies of each chromosome. So we have 2 chromatids of your mom and 2 chromatids from your dad. These guys are right next to each other, and they accidentally cross their DNA strands a little bit. Their DNA gets mixed up, essentially. And then when the cell comes to fix it, it puts this bottom part of your dad's DNA on one of your mom's chromatids, and this bottom part of your mom's DNA on one of your dad's chromatids. So you end up with this swap that happens. So this one and this one on the end are still the exact same mom and dad chromosome that you started with. But this one and this one are a little bit different. This one has your mom's AB gene, but your dad's C gene. And this one has your dad's AB gene and your mom's C gene. So this is what we mean by crossing over. The DNA accidentally gets essentially mixed together, and then when it gets separated out again, we have swapped parts of the chromosome. So pieces of genes or full genes have gone from one homologous chromosome to the other one. So we end up with a little bit different versions of the original chromosomes that we started with, right? These guys look exactly the same as our original pair, but these guys are a little bit different. They're not the exact same anymore. That's kind of what happens. That all happens during that first part of meiosis I. And then eventually these chromosomes will be separated like we were going through normal cell division. And we're going to end up with some new cells that have 2 chromosomes each. But we're in meiosis. We don't want to stop there. We want each cell to only have 1 copy of each chromosome. We want each one of these chromatids to end up in its own cell. So that's where meiosis II comes in. We basically do another round of mitosis, another round of division, but we don't copy the DNA this time. So before, we took our chromosomes, we copied the DNA, and then we separated them. In meiosis II, we don't copy the DNA. So when it gets pulled apart, we end up with only 1 chromosome in every new cell instead of 2 chromosomes in every new cell. So this is kind of hard to picture, I know, just by hearing me talk about it. So instead, I really like using diagrams to kind of show what exactly is going on here. So we start off with our original parent cell, right? We have, how did they set this up here? So we have 2 big genes and 2 big chromosomes and 2 little chromosomes. So your red chromosomes are from your mom and your blue chromosomes are from your dad in this example. So we have the big ones and the small ones. First, we go through that first part of meiosis where we're going to copy the DNA and then separate it out. So we make those copies. We then have that lining up and the crossing over happen. So remember that tetrad, that 1, 2, 3, 4 chromatid setting, they're all together. And then the inside guys do a little bit of crossing over. So this guy ends up with a little bit of dad's DNA, and this guy ends up with a little bit of mom's DNA. We then separate out those cells, or those chromosomes. So now there's 2 big and 2 small chromosomes in each new cell. Very similar to what would happen in mitosis. But then we do that again. We pull the big chromosomes and the small chromosomes apart. So now, at the very end of meiosis, we started off with 2 big and 2 small chromosomes. That was our diploid state. Now, each one of our new daughter cells has 1 big and 1 small chromosome. We've gone from being diploid to haploid. We used to have 2 copies of each big and small chromosome. Now, we only have 1 copy of big and small chromosome. We started off with 1 original parent cell. We've ended up with 4 different meiosis haploid cells. So none of these guys are the exact same as the parent cell, right? Obviously, this guy is diploid. These guys are haploid. But also, if you notice, none of these cells is identical to each other either. They're all slightly different. Some of them have the blue big chromosome, some of them have the red. Some of them have the chromosomes that have some crossing over that happened, some of them don't. So each haploid cell has a slightly different combination of genes, a slightly different combination of chromosomes. So that's what meiosis is all about, taking that original diploid cell and fully splitting up the DNA so that we have 4 haploid cells that hopefully will eventually go on, fertilize a egg or a sperm, and turn into a new zygote, which can grow into a new organism. So here's another example that's a little bit clearer. Again, we're going with kind of the big and small examples here. So we start off with 1 big chromosome from our mom and dad and 1 small chromosome from our mom and dad. All the chromosomes double up. They line up together in those tetrads. You get a little bit of crossing over that happens. The mom and dad chromosome swap a little bit of their genes, their genetic info. So we end up with a little bit different mixture than original. We then split those guys apart into new diploid cells. And then we separate those out again to make 4 haploid cells. So we split them once, they still have 2 copies of each gene. And then we split them again, so they only have 1 copy of each gene. So that is the biggest difference between meiosis and mitosis, is that we're ending up with these haploid daughter cells that are all a little bit different, right? None of these, again, are the exact same as the beginning cell, and none of them are the exact same as each other. So we start off with 2 copies of every chromosome, 2 big and 2 little copies. So each one of our daughter cells is only going to have 1 big and 1 little chromosome inside of it. And that's going to be the same for all 23 of your chromosomes as well. When your cells go through meiosis, they start off with 46 total chromosomes, but then those double up, get split apart, and then get split apart again. So each one of the final cells only has 23 different chromosomes in it. So meiosis is all about making those haploid cells. That is the main goal. It's happening inside of our gametes where we're making the sperm and the eggs. And that's why those ones are the only haploid cells in your body. All the rest of the cells in your body, all the other somatic cells in your body, blood cells, bone cells, muscle cells, skin cells, brain cells, they're all diploid. They all have 2 copies of every chromosome. Only your gametes, only your sex cells, your sperm and egg cells are haploid. So that way, when 1 haploid egg cell and 1 haploid sperm cell come together, they'll make 1 new fully diploid cell. All right. I know there's a whole lot of different steps and different parts of meiosis that I just threw at you guys there. Does anybody have any questions? Anything about this process with the chromosomes and the chromatids and all that good stuff that you're still a little bit confused about, still not really sure what's going on?
No, I got it.
Cool, cool. Thanks, Naya. Anybody else? Any other questions, concerns, confusion going on with our steps of meiosis here? All right, no problem, Ashley. All right, so meiosis, we're starting with the diploid cell and ending up with 4 different haploid cells. Mitosis, remember, mitosis is 2. They both have T. Mitosis is 2. We go through mitosis and we make 2 cells that are identical, the exact same as the original cell, the exact same as each other. In meiosis, not only are our new haploid cells not the same as the original cell, they're not even the same as each other either. They're different due to this little crossing over where we switch up the parts of the DNA. All of the haploid cells are different from each other as well. One other important thing about meiosis that goes on, that's very critical, is called independent assortment. You may have seen it mentioned earlier if you're reading through all this stuff right here, where we have these different chromatids, right? They did their little crossing over where they switched the stuff around. And then the way that they happen to line up in this case is that for this set of meiosis, the big pink chromosomes went on the left and the big purple on the right. And then the little purple went on the left and the little pink went on the right. Independent assortment is essentially a way of saying that that is not always the case that's going to happen. So independent assortment says that your maternal and paternal chromosomes, your mom and dad's chromosomes, don't always separate to the same side of the cell, right? We have 23 different chromosomes in humans, and independent assortment says that they're not going to be separated the same each time. So like in this super simple example where we only have 2 different chromosomes, a big and a small chromosome. In one case, you could get both of the blue on the left and both of the red on the right. So this will go through meiosis I and split up once, and then go through meiosis II and split up again. And so you could end up with a cell that has, or 2 of our 4 could have a big and little blue, and 2 of our 4 could have a big and little red. Or what could happen is that the big blue could line up on the left and the big red on the right, but then it's switched for the other ones. The small red is on the left and the small blue is on the right. So then these guys will go this way, these guys will go this way. And this time we ended up with 2 cells that have a big blue and a small red, and 2 cells that have a big red and a small blue. So all independent assortment is saying is that it's always random which side of the cell that the genes will be pulled to. There's no like set way that they're going to be lined up every time. So you're always going to have kind of a random assortment of gametes. You're not going to end up with the same gametes every time when you go through meiosis. So this is one of the ways that not all of those haploid cells look the same, right? So each time we go through meiosis, in this case, there's a pretty limited number of possibilities we can have since we only have 2 different chromosomes. But again, in humans, we have 23 different chromosomes. So there are, I don't even want to do the math, but that means there are thousands and thousands of different ways that they can be lined up and separated on either side of the cell. So independent assortment is one way that we can end up with lots and lots of different gametes, gametes that are not the same as all the other gametes around them. So the other way, like I was talking about earlier, is crossing over. So remember, crossing over, they don't show it in this picture, but that's the one where they swapped a little bit of DNA with each other. Crossing over also makes it so that your haploid cells are a little bit different from each other as well. So between crossing over and independent assortment, we can get a huge amount of different combinations of genes in our gametes, in our sperm and egg cells. In biology, we call that genetic diversity. Lots of diversity of different possible genes in your sperm and egg cells. And in science, we really like genetic diversity because genetic diversity is good for populations. The more diverse genes that you can have passed on from generation to generation, the more different combinations and possible new traits and new possibilities for survival that you could develop. And so this is why you don't look exactly like a complete 50-50 mix of your parents, and why you and all your siblings don't look the same either, right? If you've ever seen like a really big family of like 5 or 6 kids, sure, they all share the same overall gene pool, so they all look kind of similar to each other. But assuming that there's no like identical twins or triplets, none of them look exactly the same, right? And none of them look exactly like their parents. And that's all because of this crossing over and independent assortment. We can create a huge amount of different genes, different gene combinations in our gametes, so that when that sperm and that egg come together to start making the zygote, making the embryo, and eventually the baby, there's almost always going to be a different set of genes in that sperm and egg every time. So again, you probably don't look exactly like any of your siblings unless you're a twin. If you see other families, they don't look exactly like each other or exactly like their parents. It's all because of this crossing over and independent assortment. So we get this huge variety of different combinations of chromosomes and genes and gene types and stuff, which is what allows for the huge amount of diversity we see on Earth, right? Besides identical twins, no two people on Earth look exactly alike, right? It's all because of the different combinations of genes that they inherited from their mom and dad, and crossing over and independent assortment. So switching a little bit of DNA between the mom's genes and the dad's genes, and making it so that the chromosomes will line up in different ways each time, leads to having a huge amount of genetic diversity in our gametes. Ooh, good question, Ashley. So the main way that you get identical twins or identical triplets is that, I forget exactly what happens, but essentially one sperm and one egg come together, and while they are coming together, the egg essentially duplicates, right? So instead of the sperm and egg come together, make one cell, and then that cell goes through mitosis and makes 2 cells, and then 4, and then 8, and so on and so forth. Instead, what can happen is the egg becomes duplicated before after all the DNA has gotten in there, but before it's ready to start going through mitosis. And so that's how you end up with identical twins, because it was the same sperm and the same egg that came together. But instead of just developing into one embryo, they started developing into 2 embryo. At some point, they completely copied all the cells, and each set of cells started turning into its own human. And that's where identical twins come from. So identical twins have the exact same genes in their cells. Genetically, they're the exact same as each other, But they're different people because of that egg duplicating and splitting up. On the flip side, when you get fraternal twins, so twins where either they're not identical or you get like a boy and a girl twin, that usually happens because the ovary, instead of just releasing one egg at a time like it usually does, it accidentally released two. So instead of one egg getting fertilized by one sperm, two different eggs got fertilized by two different sperm. And that's how you end up with non-identical twins. Sure, no problem. Good question. So yeah, so besides identical twins where the sperm and egg come together, but then they duplicate, everybody else looks a little bit different from their parents and their grandparents, and it's all because of this mixing and changing up of their genes. So, in kind of summary of comparing between mitosis and meiosis, which is something that you guys are also going to do for your homework today, which we'll talk about in a second, they both copy the DNA once, right? So in both cases, we make a completely second full set of DNA. In mitosis, we only divide once, where we go through all 4 steps of mitosis, split that one cell into 2 brand new identical cells. But in meiosis, we go through 2 splits. Essentially, we go through 2 rounds of division, 2 rounds of mitosis. So we go through prophase, metaphase, anaphase, and telophase, and then we do it again in meiosis. In mitosis, there is no crossing over. There is no case where the chromosomes, the tetrads, all line up super close and get crossed over. But in meiosis, we do have that happening. In mitosis, we end up with 2 identical diploid cells. So 2 exact copies of the parent cell. They both have a full set of 2 copies of every chromosome. In meiosis, okay, so remember, mitosis is 2. MITOSIS, TWO, 2 new cells. Meiosis, we don't get 2, we get 4 cells. Each of these 4 cells is haploid. It only has 1 copy of each chromosome. And they are different from the parent cell, and they're different from each other cell. Thanks to crossing over and independent assortment, we end up with completely different daughter cells. So they're very different from the parent cell because they're not diploid anymore, they're haploid. But they are also different from each other. No 2 daughter cells looks the same. And finally, why do we do mitosis? We do it for growth, to create new cells in order to grow. We do it to repair cells that get damaged, and we do it to replace cells that die. So some cells just naturally don't live very long, like blood cells have to be replaced after a couple weeks, and skin cells have to be replaced after a couple months. And it's thanks to mitosis that we can do that. So mitosis has lots of roles. Meiosis only has 1 role, and that is to produce gametes. So the full reason why we do meiosis is so that we can get those haploid gametes, so that eventually when they come back together, they'll form 1 full diploid cell. So meiosis only happens with the gametes. No other cells in the body deal with meiosis. Only the gametes. Everybody else goes through mitosis. Everybody else is just making copies and copies and copies of themselves. All right, so then finally, this isn't super important, so I'll kind of breeze through it. Eventually, cell division will be stopped, sometimes by, if there's too many cells around, then they're like, oh, okay, we're good. Don't need to make any more cells. However, there are certain cells that something goes wrong and they don't stop dividing. They just keep going and going and going and going and going, making more and more and more and more copies. And we call those cells cancer cells. So cancer is what happens when cells stop behaving and start just making more and more and more copies of themselves. So the reason why people who have different types of cancer often develop tumors in their body is tumors are just big lumps of cells. They're big groups of cells that have just not stopped dividing. The body has tried to tell them, hey, stop, don't do that. We don't need any more. But something has gone wrong inside the cell, and it is no longer listening to those stop commands. So it just grows and divides, grows and divides, grows and divides over and over and over again. That's what gives us tumors. So benign tumors is what we call it when they're growing in places that they're not really causing any damage to anything around them, and they're not growing very fast, and they're easy to get rid of. Malignant tumors are when they are doing damage, they are growing in places that's causing a lot of issues, and maybe they're hard to get rid of, or you get rid of them, but they come back. So the main reason why people die of cancer is it's not like some poison that builds up in their body and causes them to eventually die. They die because all of this cell growth, all of these tumors that grow, make it too hard for some other super important part of the body to keep working. So maybe the lungs, or maybe the heart, or maybe the brain. As the tumors grow bigger, they put a lot of stress on that organ, and finally, they could eventually cause that organ to stop working. Most of the organs in our body, we need them in order to survive. And so if tumors grow big enough and they shut down those organs, that's what causes people to die from cancer. And so it's not like some thing in the air or some disease that can be passed from person to person. All cancer is really is just cells that will not stop growing and dividing. They won't stop going through mitosis, no matter how much the body tells them to stop. And eventually, there's just too many cells, and they overtake some especially essential part of the body. So there's all sorts of different gross tumors that can grow in all sorts of different areas of the body. But again, they're just thousands and millions of cells that just grew and divided and grew and divided and would not stop. Blah, blah, blah. I'm not going to super go through the different types of cancer. Basically, the names for different types of cancer come from where they're found on the body. So like a carcinoma is when it starts off as a skin cancer or the tissue that like lines the internal organs. Sarcoma begin in like muscles and blood vessels. Leukemia happens in bone marrow is normally where that begins, so on and so forth. So basically all of different treatments for cancer revolve around trying to make these cells stop growing. So like chemotherapy and radiation treatments are specialized treatments that target growing cells and try to interrupt something in those cells and get them to stop growing. Sometimes if you can find the massive tumors and easily take it out, that could be enough to take out all the growing cells so that you're left over with the normal healthy cells. Sometimes you can get new blood or bone marrow to replace the one that is growing all the tumors. So as of yet, there's no cure for cancer. Scientists have not figured out a especially great way of getting cells to stop growing when they're not listening to the body. But obviously, there is a ton of research going on to try and figure out how to get cells to respond to those stop commands from the body. All right, so sorry to leave it on a little bit of a downer note, but I know cancer is something that a lot of people have had affect their lives in some way, either themselves or a friend or family member has gone through it, and it is directly related to this cell division. So cancer is only due to cells that won't stop going through mitosis. They won't stop growing and dividing, even when in normal cases, they should have stopped a long time ago. All right. So you don't have to worry too much about the stuff about the cancer. Basically, just know that cancer is caused when cells don't stop growing and they just keep dividing and dividing and dividing. All right. Does anybody have any final questions about meiosis, mitosis, different parts of cell division, the differences between them, anything that I've talked about so far from today or even from Tuesday?
No. You covered everything I was asking from earlier.
Okay, awesome. Good to know. All righty. So like I was mentioning earlier, your homework for today has to do with that mitosis versus meiosis kind of stuff. Unfortunately, it's one of those homeworks where I couldn't figure out a really good or really efficient way to set it up in Google Forms. So you're just going to have to fill out a simple little Google Doc table. And essentially, all you have to do is fill out these boxes right here with a sentence or a phrase that best helps describe this question for mitosis and for meiosis. So there are, let's see, 1, 2, 3, 4, 5, 6 questions. Why do cells go through mitosis and meiosis? Where does this type of division happen? How many times does the cell divide when it goes through mitosis versus meiosis? How many new cells are produced at the end? Do we end up changing how many chromosomes are in the cell in mitosis and meiosis? And what is the overall final result? What do we end up with at the very end of the mitosis process and the very end of the meiosis process? I'll go ahead and tell you guys now, since you guys are here in the class with me, is that this answer will essentially be kind of a combination of these two answers. So I want to know, when all is said and done, when you've gone through one round of mitosis or one round of meiosis, what do you end up with? What do your final products look like? So again, you don't have to write like paragraphs and paragraphs in here. Just if one sentence or one phrase or one word best describes what's going on in mitosis versus what's going on in meiosis, then that works fine for me. All right. Anybody have any final questions on the assignment for today or meiosis and mitosis in general?
No.
All right, what's going on Monday? So remember, since this is a night school class, we do still have normal school day on Monday. So I think it's just a simple question about meiosis and mitosis. So make sure to check back in on Monday and answer that question to earn your Monday attendance credit. Don't forget that the midterm is going on now. I know a couple people have already turned it in, which is great. If you haven't turned it in yet, that is totally fine. Remember, you have until the end of the day on Friday to get it turned in. So you have the rest of the night this evening and all day tomorrow to work on it and get it turned in. So again, take your time, read through the questions, make sure you understand what each one is asking, and feel free to reach out to me for help. I can't necessarily help you answer the question, but I can help you understand the question and understand what sort of answer that it is looking for. And then finally, don't forget, at the end of this class, or I should say after this class, at between 6:35 and 8 p.m., there is a science and math tutoring session going on in the advisory class that Ms. Mota and I host. So the link for it is here on the main Biology B page. If you scroll down a couple posts, you can find it. So if you have any science or any math questions or tutoring help, either for a science or math class that you are in, or the biology star or the algebra star test, feel free to hop on. It'll be open the entire time between 6:30 and 8. And we will be more than happy to answer your questions and help you out with anything that you need relating to science or math.